Today we will learn about secretine. In previous video, we were talking about gastrointestinal hormones, and this was divided into three divisions, main divisions, depending upon their chemical structure. That was secretine family, then gastrin family, secretine family, and others. Today we will talk about secretine family, in which secretine is the main hormone. This hormone was discovered by Belicis and Starling in 1902. This was the first hormone discovered in the history. Secretine is secreted by S cells present in the crypts of the mucosa of upper part of the small intestine, duodenum and jejunum, and it is also secreted from other sites. It is secreted as a proenzyme which is converted into enzyme. The proenzyme is known as prosecretine, and it activated by gastric acid and salts of fatty acids into its pure form, that is secretine. After secretion, it is liberated into the portal circulation, and then from portal circulation it will go into the systemic circulation and then to the target tissue, that is pancreatic tissue, to exert its effect. Structure: it is a polypeptide with 27 amino acids and resembles that of VIP and GIP glucose structures. Resembles with the hormones of other hormones, that is VIP, vasoactive, inhibitory, polypeptide and GIP. Then mechanism of action. Cells, we know that these hormones act through receptors. So, on pancreatic centroacinal cells have receptors for the secretine. These receptors are present on the plasma membrane, and when secretine binds to these receptors, it stimulates adenyl cyclase activity and converts ATP to cyclic AMP in the pancreatic duct cells. Cyclic AMP acts as a second messenger in intracellular signal transduction, resulting in secretion of alkaline, watery pancreatic juice poor in enzyme. While CCK, cholecystokinin, pancreozymin causes increased flow of pancreatic juice rich in enzymes. Secretine also increases water and bicarbonate from duodenal Brunner's gland. It modulates water and electrolyte transport in pancreatic duct cells, liver, cholangiocytes and other cells. It also plays a role in the vasopressin independent regulation of water reabsorption from the renal tubule. Secretine is also present in meganocellular neurons of the paraventricular and supraoptic nuclei of the hypothalamus, and along with neurohypophysis. During increased osmolality, it is released from the posterior pituitary. In the hypothalamus, activates vasopressin release. It is also essential for the central effect of angiotensin II. Secretine and its receptors are present in diabetic nuclei of discrete nuclei of the hypothalamus, including the paraventricular and arcuate nucleus, which are the primary brain sites for regulating body energy homeostasis. So, secretine is also secreted from other sites. When the volume of pancreatic juice increases, its chloride concentration falls and bicarbonate concentration rises. In a small pancreatic ducts, bicarbonate is secreted but reabsorbed in the large pancreatic duct in exchange for chloride. In small ducts, bicarbonates are secreted, but this is reabsorbed in large pancreatic ducts and for chloride in exchange of chloride. Functions: increases secretion of pancreatic juice rich in bicarbonate, watery and alkaline pancreatic juice, produces enzyme rich juice by the pancreas, stimulates alkaline bile secretion. On the secretory side, it increases secretion of pancreatic juice rich in bicarbonate and water, and also stimulates CCK-PZ, cholecystokinin, pancreozymin, to stimulate enzyme rich pancreatic juice. and also stimulates alkaline bile secretion. On other hand, it causes decreased secretion of gastric acid and decrease or increased contraction of pyloric sphincter, so delays gastric emptying. Stimulates secretion of bicarbonates and enzymes from the pancreas and alkaline bile, while inhibit gastric acid secretion and motility of stomach. Factors influencing secretin secretion is acidic chyme and products of protein digestion, that is peptide, dipeptide and amino acids, and causes its increase, while alkaline chyme present in duodenum and jejunum causes decrease in its secretion. Now, gastric inhibitory polypeptide. Gastric inhibitory polypeptide GIP is also known as glucose-dependent insulinotropic polypeptide produced by K cells present in mucosa of duodenum and jejunum. Its secretion is stimulated by the presence of glucose and fat in duodenum and jejunum. Function: decreases secretion of gastric acid and gastric motility, while stimulates beta cells of isolate of Langerhans of pancreas to increase secretion of insulin. It decreases secretion of gastric acid and gastric motility and decreases the motility and gastric acid secretion and stimulates beta cells to produce more insulin. Vasoactive intestinal peptide. It is present in the GIT, released from jejunum in response to fatty meals. It is also present in the blood, brain and ANS. Functions: relaxes smooth muscles of intestinal intestine, gut, including a sphincter, blood vessels and genitourinary system. It relaxes smooth muscles, so peripheral blood vessels are dilated. It decreases gastric motility and stimulates secretion of electrolyte and water, increase the action of acetylcholine on salivary glands, causing profuse secretion of watery saliva and relatively low content of organic material and protein. Glucagon is secreted by gastric mucosa and duodenal mucosa, but it is different from alpha cells of glucagon secreted by alpha cells of isolate of Langerhans. Function: it reduces glucose level in hyperglycemia. So, these were, that is gastrin, cholecystokinin, pancreogymin, secretine, VIP and GIP are major or principal gut regulatory hormones. Now, other hormones are MO cells, motilin secreted by MO cells in the stomach, duodenum and colon. These regulate gastric motility by causing contraction of smooth muscles of intestine. Neurotensin secreted by mucosal cells of ileum in response to fatty acids and inhibits gastric motility. Peptide P secreted by mucous cell of jejunum in response to fatty diets inhibits gastric motility and gastric acid secretion. Substance P formed in the endocrine cells and nerve cells in the GIT causes increased motility. Substance P formed in endocrine cells and nerve cells in the GIT causes increased motility. That is, we have seen that first in neurotensin and peptide P are inhibitory, gastric motility inhibitory, while substance P formed in the endocrine cells are and nerves and GIT are causes increased motility. Then gastrin releasing peptide causes increased gastric secretion. Then ghrelin, a peptide hormone from stomach and liver. This is often known as hunger hormone because it is formed in large amount, it is found in high level in starving patient or fasting individuals. Its agonists are being used in the treatment of anorexia and loss of appetite. Amylin controls glucose level and gastric motility. Oxyntomodulin controls acid secretion. Then somatostatin, this is produced by delta cells of islets of Langerhans, pancreas and GIT mucosa. It is inhibitory hormone and inhibits the secretion of gastrin, VIP, GIP, secretine, motilin, insulin, glucagon, pancreatic juice and gastric acid secretion. It inhibits GIT motility and contraction of GIB, that is gallbladder. It inhibits absorption of glucose, fatty acid and amino acid. All nutrients are absorption are inhibited by this. So this is this is in nutshell about gastric intestinal hormones. As I have said that these 3 or 4 gastrin, pancreogymin, cholecystokinin, pancreogymin, secretine, VIP and GIP are main, but others are also present. They also have some function on the GIT secretion and motility. Thank you very much.